Chapter 1, Section 3, Cell Size and Morphology. Now, one of the things we need to be able to be prepared to do is to discuss the shape of microbes. Shape is dictated by, as I said in the previous lecture, shape is dictated by the genes, okay? The genes call for certain structural proteins to be made, certain cell wall components to be made, and which are made and how they're structured, how they assemble will dictate shape and dictate size. The majority of microbes are going to be between 0.5 and 10 micrometers in length, okay? Remember, micrometer, that's a millionth of a meter. Now, not all of them are going to be quite this small. Some of them, Euploiscum fissionisonium, Thymargarita, Nembezians, these are actually big enough that you can almost see with the naked eye. You know, from what I've been told, some of these actually look like, almost the size of grains of sand. Somebody once claimed they saw some that looked like really, really small, pieces of rice. Don't know about that. Haven't seen that personally. Keep in mind, 0.5 to 10 micrometers in length is where you're going to find the majority of most prokaryotes. Eukaryotes, 5 to 100. So, about 10 times, like I said, previous lecture, 10 to 100 times bigger, depending upon, again, which prokaryotes, which eukaryotic cells you're referring to. So you go and you look at these relative sizes. E. coli is one we're going to play with a lot in lab. And you go and you start looking at some of these. Thymargarita, Nembezians. It's 100 million times. This is one of those they supposedly claim can be about the size of a grain of rice. Haven't actually seen it. I've seen pictures of what was purported to be, a prokaryote. I don't know. We'll see. Maybe one day. But things can get big. Okay, so not every bacteria is going to be small. These are exceptions to the rule. So we're going to see in lab, though, just to be able to see the ones that we're going to play with, E. coli, Staph aureus, strep, things like that, you're going to need a microscope with 1,000x magnification. E. coli, whereas the ones on the previous slide were 10, 100,000, 100,000, millions bigger. There's also going to be some like Mycoplasma Pneumoniae, which is a pathogen you find that's really the bane of existence of a lot of hospitals. Look at it. It's 0.0025 times smaller than E. coli. And as we're going to see in later chapters when we're talking about this guy, it's also doesn't have a cell wall. And things like that make it very hard to get rid of. Turns out a lot of our antibiotics are against cell wall composition, synthesis, and formation. So when you look at the shapes of many of these microbes, as I said, are going to be dictated by the genes. Here's Paramecium. Paramecium is a single cellular eukaryotic protist. Epolipsicium cell is a bacteria. Notice, it's about 10 times larger than a eukaryotic cell. Okay? This is the exception to the rule of being extremely small. A lot of times students will ask, why do the microbes have to be so small? Why can't they be bigger? Well, it's all about surface area. You have this cell. Now, if the cell's huge, trying to get things from one side of the cell to the other or somewhere in between, it's going to take a lot of time. Because remember, things have to diffuse through these cells because they're prokaryotic. They don't have organelles to help move stuff around, to store stuff in. So they constantly have to bring in their nutrients from outside the cell and allow it to slowly diffuse across from one side to the other. Can only get in so much. But what they've figured out, scientists have figured out, that evolution has selected for smaller cells because by being a smaller cell, surface area to volume gets maximized. You have one big cell, radius of 2. Got to remember, your volume, 4/3 pi r cubed for, that's the volume of a sphere. Volume, if the radius is 2, the volume is 33.5. Surface area, 50.3. That's a lot of surface area. That's a lot of volume. The surface to volume ratio, though, 1.5. Ugh. Trying to get stuff from here all the way across, diffusing across, it's going to take a lot of time. The stuff might get used up by other metabolic activities before it can get over here to where it's needed. Whereas if you just cut the volume in half, cut the radius in half, radius of 1, surface area 12.6, oh good lord, that's like 4 times less surface area. Volume 4.2, oh my, that's like 8 times less, 7.5 times less. But look at this, the surface to volume ratio doubles. It means it's now easier if you have some nutrients that are being absorbed into the cell from here, they can diffuse through the cell at a faster rate. So it gets dispersed through the cell quicker. Dispersion through the cell here, slow. It's not going to happen under a time frame that's good for the cell. Now, there's some nomenclature you're going to have to use, okay? It's basically from here on out. These are the words you have to use because nomenclature, what words we use in science, that's coin of the realm. That's how we do stuff. Coccus, bacillus, spirillum, spirochete. Spheres, rods, spirals. These are how we describe the shape of an individual cell, an individual bacterial or archaea cell. Are they grouped, clustered? That's where you get ideas of things like diplococci, streptococci, staphylococci. Strepto, chain, diplo, it's 2, tetrads. You're also the staphylo, that's a cluster. Strepto is Greek for chain, staphylo is Greek for cluster, a grape, like a grape cluster. So we need to start using these terms. Streptococci, strepto, chain of spheres. Streptobacilli, plural of bacillus is bacilli. Chain of rods. Diplococci, diplobacilli. It's 2 spheres or 2 rods that are joined. Always 2 together. So when you look at them, here's the artist, here the rendition of what's shown here in a black and white microscope picture. Cocci, sphere-like. Here, this is a sphere that's dividing into two, but here you see these sphere-like. They're not true spheres. Bacilli, these rods. Spirillum, the spirals, not always going to be tight spirals. They can take on more of a comma-like shape or some bent. So you take these bacilli, lengthen them a bit, and put a bend or two into them. That's spirillum. Now, if it takes on a true corkscrew, that is a spirochete. And yes, that is a single-cell organism. Rickettsia. It literally looks like a really tightly wound corkscrew. And then you can get in some other weird, quote-unquote, weird shapes. You know, budding appendages. You know, this is a single-cell organism, and this is a stalk, a hyphae that it would use to adhere to things. Or it can get long, these filamentous. These are long strands of almost a streptobacilli, except it's hundreds and thousands of them long. And that's just how they grow.